Okay. Hi everyone, how you doing? Okay, so my TKI is called Imatinib. Okay, so the context of this project, so the PBD IV, first of all, is I like to call it 6 MOB or 6 MOB, and it's for Imatinib, which is also referred to as KIT, which is a cancer treatment drug, which is a tyrosine kinase inhibitor. And it blocks the activity of abnormal proteins, which are the tyrosine kinases. So imatinib was the first FDA-approved TKI, and it was used to treat gastrointestinal stromal tumors, or GISTs, and CML, which is chronic myeloid leukemia, and other soft tissue carcinomas. And so just the overview of the whole experiment. So the mutation for KIT is called V654A, and the wild type is KIT, and it's used to find the potential inhibitor. So the whole goal of the experiment is to find the potential inhibitor and narrow it down to the best 10 compounds. And so the significance and the importance of this is that research has enabled driver mutation identification in 99% of GIST patients. So what that means is in 99% of people who have gastrointestinal stromal tumors, they've been able to find what caused it. Where is that mutation? And so being able to identify this mutation within KIT, it leads to, treatments for things like GIST and CML. And so the prospect of this experiment, it's okay. It's just that finding potential inhibitors has proven to be pretty difficult. Okay, so details of the project description. So what happens is that KIT is protein prepped and it goes through homology modeling, and it's performed so that the wild type KIT can be tweaked into the mutation. And so that mutation, V654A, it undergoes high throughput virtual screening, extra precision and standard precision docking. Not in that order, but it like goes through these steps to filter out and to find the best potential inhibitor within the zinc library. And so there's this thing called ADME, or ADMET is normally how it's referred to now, but it's absorption, distribution, metabolism, excretion, and toxicology. And these are all characteristics in pharmacokinetics. And they basically do a prediction using this to prevent further unnecessary testing that they would otherwise not actually have to do. So the objective, like I said, find at least 10 desirable inhibitors. The goal was to find at least 10 for KIT, the mutation from the testing. And so some key scientific problems in this, finding potential inhibitors has proven to be difficult. As I said, it actually used to be considered impossible, but now it's not anymore. And there were issues with drug resistance and toxicity, and that's proven to be a challenge for kinase inhibitors within cancer treatment. Okay, so the research approach to all of this, the methodology. Figure 1 on the right is the virtual screening workflow, thanks to the bioinformatics student, Rithika. And it's just, this is the method that they've used to identify the best competitive inhibitors. So it starts out with the zinc library, getting 6MOB from the open public database, and then it goes through its first ADMET filter, and then the first type of glide docking is HTVS, and from there they select the top 10% of competitive inhibitors to move on to the standard precision docking. Then after that happens, they take the 10% of those to move on to the extra precision docking, which kind of is like a filter for everything that was able to slip through before. And then, they analyze the structure of it and pick the top 20 compounds from that portion. And then they use a program called Desmond, and they just simulate and analyze the compounds and everything. Then it goes through another ADMET prediction, and from there it says top 8, but they really selected top 10 compounds, and see if there even is 10 that can be selected. So feasibility analysis of this, So virtual screening, as opposed to being in the lab and experimenting, it's cost effective, it's able to be repeated if needed, and it's efficient and it saves time. And using the ADMET prediction, again, avoids unnecessary testing and trials that would otherwise not need to be done. And filtering the top 10 to 20 compounds or so, it ensures inhibition potential and, basically selecting the best of the best. Okay, so the originality, the innovation. This is kind of a novel type of experiment because in the past few decades, not a lot of research has been done with virtual screening of imatinib specifically. And most of the experimentation that was done was primarily in the early 2000s. And what makes this hard is that certain gastrointestinal tumors are resistant to imatinib, which, hence the fewer studies. And the Zinc database allows ease of access to chemical compounds for virtual screening because it's open, it's public, it's free. And the combination of the three filtration methods, the HTVS, the XP docking, and the SP docking, they serve as multiple filters. They weed out any false positive docking scores that standard precision would otherwise allow through. And so the research plan and expected results. So, again, like I said, so the whole plan is to undergo all of these virtual screenings, HTVS, SP, XP, in that order, choose the top 20 compounds from those, then use Desmond and analyze it, the ADMET prediction, and then choose the top 10 potential inhibitors, hopefully. So the expected result was that there would be 10 potential inhibitors. And so I'd like to thank Rithika Pathi. She's the bioinformatics student who I obtained a lot of this data from. And that's it. Any questions? Yeah.
Are you actually able to find any inhibitor hits?
Yeah, so I did look into Rithika's paper. After everything, all of the filtration and docking, she was actually only able to obtain one hit. Like after everything, there was only one potential inhibitor that was good enough, I guess. Yeah. What's up?
Why is it difficult to choose a potential inhibitor?
So it's difficult because research has shown that it's kind of like certain amino acids that interfere with imatinib's ability to interact directly with kinase inhibitors and everything. That's basically what that's about. What's up?
Did you talk about the signaling at all, the cascade?
No, there wasn't a lot to pull from this, only because it's such a difficult inhibitor to work with, basically. What's up?
In regards to the novelty of these experiments, is this specific for only this type of drug?
Yes.
Because I feel like in silico methods for large pharmaceutical companies now is kind of becoming the standard.
Oh yeah, it's definitely becoming the standard as in, recent years, but just as a whole, imatinib, the whole history of it, not a whole lot done.
Question. Okay, so actually the critical part is really the final report. Experiment part.
Yeah.
This virtual screening is really identify the candidate. Then in your final report, you want to discuss with them found the IC50 for this top 5 compounds and why have new. Then you discuss the method we use, which will evaluate go max kind of toolbox to monitor the AT reaction. And then for each protein, you have the bioconcentration. One discuss what bioconcentration you use. So we have the body control, nitrogen control, and 3 with the inhibitor. The legal one is called IC50. And then after we measure the IC50 for the 6 compound, including the random compound,
Mhm.
we want to propose how to interpret that, what kind of the result was suggest we identify the compound good. So yeah, so that major portion one working in the other part. It's good. Okay.
Thank you.